Good morning. It's a pleasure to be involved in this workshop on the role of ion gradients and pumps in neuronal activity. And thanks to Genna for thinking of the idea of having this conference. As you can see, I'm not in the same place that the conference recording actually took place in, and that's because the first few minutes of my presentation were garbled. So I'm adding on to that presentation here. So I'm going to share my screen now and join in the presentation. So we are, as a research group, are Genna Cimboluk, Ron Calabrese, Ricardo Arazo Toscano, a graduate student, and Parker Ellingson, and we have been working on modeling and experiments to indicate a role for the sodium-potassium pump in the generation and regulation of bursting activity. So my lab for a long time at Emory has been studying the central pattern generator that controls heartbeat in the leech. And this is a distributed pattern generator that occurs in distributed throughout the ganglionic chain of the leech nerve cord, and the interneurons that comprise it are distributed in the first 7 ganglia. There are within the 3rd and 4th ganglia pairs of interneurons that form half-center oscillators, where there are 2 neurons that are mutually inhibitory, and these new neurons produce alternating bursting activity. And there are 2 different ways we can record from these, either with an extracellular kind of loose patch electrode shown at the top here, or with intracellular sharp microelectrode recordings. So these ganglia, when isolated, will produce half centers. And if we use these ganglia down here, when they're isolated, then we have individual neurons that are synaptically isolated. Our thoughts about how the pump contributes to bursting in hard interneurons that make up these half-center oscillators began with a study a long time ago by Anneliese Tobin, in which she was looking at the modulator myomodulin. And as you can see, here's a half-center oscillator producing rhythmic activity in an isolated ganglion. And then if you put on 1 micromolar myomodulin, this speeds up the rhythm of bursting. And it increases the mean, the max, and the minimum spike frequency also. So the period decreases. So this was, her goal was to figure out which currents were modified by myomodulin to cause this change in period and spike frequency. She also noticed in experiments like this that myomodulin enhances the inherent burstiness of the hard interneurons. And here you'll see at the top a half-center, 1 recorded intracellularly, 1 extracellularly, 1 member. And you can see that at this point, she put one of the interneurons into voltage clamp at 50 millivolts. There's some escape spiking here, minus 50 millivolts. And you can see that the other cell continues to burst, but its burst rhythm is disrupted somewhat. It's more irregular, it's prolonged, and it looks a little weaker. Now, if you do this same experiment in the presence of 1 micromolar myomodulin, then when you break the half center with this voltage clamp protocol, then you see that the opposite cell does not perturb its rhythm at all. In other words, we've enhanced the inherent burstiness of that neuron. And this is shown here that myomodulin decreases the coefficient of variation in the period here versus here, and it also decreases the burst duration here versus here.
What causes this? So Anneliese Tobin, who did this work, and you can see the reference below, looked at all kinds of different currents in these cells to see what was modified. And she found that the H current, which is active during the inhibited phase of the rhythm, is enhanced by modulation. And that's shown at the left. She also found that using voltage ramps, that there was a reduction of an outward current in myomodulin. You can see that in the right-hand panel in magenta. So in myomodulin, the ramps shift down, and there's not a change in slope of the ramp current, indicating that there is no associated conductance. And then when Wabane is used, which blocks the pump, then you see that myomodulin doesn't cause this shift. So that was the first indication to us that what myomodulin does is not only enhances H current, but also decreases the outward pump current. And in leeches, there's a long history of studying pump in sensory cells, and there serotonin and various other modulators do exactly the same thing. They lower the pump activity. So this started us to look at this, at the pump and its role in rhythm generation. Okay, so here is now a model of a half-center oscillator that incorporates the pump in it. And you can see that the two cells are alternating, and then you see the role of the H current in magenta, the pump current in green, the sodium concentration inside the cell, and the IP, which is our persistent sodium current. So those are the traces, and what we're doing here is we have all these different currents shown at the top, and they're synaptic currents, of course, associated with the inhibition, and then the internal concentration of sodium is calculated by looking at the contributions of various different currents to the sodium concentration. So H current, part of it is sodium, the persistent sodium current, the fast sodium current. There's part of the leak current that's sodium, and then there's the pump current. And our pump model is one that has no time constant. So it instantaneously responds to the internal sodium concentration and increases its activity. So really, the slow variable in the system that we have is the buildup of the sodium concentration. And you can see here that during a burst, the sodium concentration builds up and the pump follows that buildup. Meanwhile, one of the big contributors to this sodium concentration is this large inward persistent sodium current. So what's happening here is that the pump current is helping to terminate these bursts. And it's also providing a strong hyperpolarization here that activates H current.
Ron, so, I have a question.
Yes.
Given the range that your internal sodium concentration is varying over, is the pump current basically going off all the way to max, or is it only exploring a small part of its dynamic range? I wonder, you know, like what's the...
It's not going to max
Okay.
at all.
So even at its peak, only a fraction of the pump is activated.
That's right.
Yeah. Okay.
That's correct. We are not in this state. There are states of the system where the pump will saturate, and Genna will talk a bit about that. But here, the pump is not saturating at all.
Okay.
And so, all right. So now, so I borrowed this technique from the Martyr lab. This is what's called a currentscape, and it shows you one member of the half center oscillator and then gives you the percentage of each current that's flowing at any given point in the cycle. And the outward currents are shown at the top and the inward currents are shown at the bottom. And here is this persistent sodium current in orange. You can see that it makes up an enormous percentage of the inward current and contributes a lot of sodium. Here's the H current. It builds up during this inhibited period. And here's slow calcium current. It's helping to sustain this burst. And here's the fast sodium current. The light blue is the leak, and the leak, as you might imagine, changes direction according to the waveform. But what's interesting here is that even the inhibition changes direction because the pump drives the membrane potential below the reversal for inhibition. So here's inhibition. And the main outward current turns out to be the pump, with a strong contribution from the leak current and the various potassium currents. So that's the way the model kind of comes in, and I thought this was a nice way of displaying it. Thanks to Eve Martyr for.
Ron, I have a question. Does the pump also change the sodium reversal significantly?
No, not really, because it doesn't change that. It doesn't allow the internal concentration of sodium to change very much.
Okay.
So
I also have a question that is so basic. I should clarify this right now. What you call pump current, is that the sodium, or is that the net sodium and potassium?
okay, what we call the pump current is the difference between how much sodium is thrown out and how much potassium is in. But when we calculate the sodium concentration, we have to use 3 times that. That's the equation has 3 times in it.
So for the sodium dynamics, you use the actual number of sodium ions being pumped, whereas for the electrical consequences, you use the net 3 sodium out, 2 potassium in. That's 1 charge.
That's correct.
Okay, good.
That's the way we've set up the model. Okay, so
Sorry,
now here's
one more.
yes.
Going back to the currentscape. When I see, for example, that the pump looks like it's the largest right after the burst terminates, is that an absolute measurement or a relative measurement?
That's the relative measurement.
Relative, okay. Thanks.
That's what the currentscapes give. All right, so here is something that Parker did, which I think is really fantastic. So here is a map of pump current, pump maximal current. So we are hypothesizing that myomodulin changes the max pump current. That is the number of pump sites available. and the maximal current of H. Remember, it's those two things that are modulated by myomodulin. And so in this thing, we see an area where the half center is asymmetric and it's just not functional. And that's shown in green here. And then there's another area where the bursts are highly variable and they don't conform to anything that we see in biology. But then there is this nice pathway in blue through this parameter space where the system is what we call functional. That is, they're nice, symmetric, alternating bursts, and they have the right period and they have the right spike frequency to be considered realistic. And so now I'm showing you that passageway here, that passageway shown in terms of the period that's expressed. And so if you were, if you wanted to modulate this period, you started at a particular pump level, you would get a very limited range of periods you could change just with age. Similarly, if you want to change period by changing the pump, you would have a limited range. But if you change them simultaneously, then you move through this passageway, let's say along this path of black, and you could get a larger range of periods that you could realize by just modulating one. And then what this black line was fitted to move to the data. So here then is the model in magenta and the living neurons in blue, and Daniel Quay did this by adding different concentrations of myomodulin to the system. And I'm not showing you the control. The control sits here on this curve. We also did it when the H current is partially blocked by cesium. It sits here in this green spot. This is 1 micromolar myomodulin, this is 10 micromolar myomodulin, and this is 100 micromolar myomodulin. And here are, so that's how this pathway was computed to fit through this parameter space. So if you co-modulate these two variables along that path, then you can have a maximal effect on the period. And Genna will talk a little bit about spike frequency when he
Ron, what made you choose the path so close to that upper bound where you're really close to kind of falling out of synchronous?
What made us choose that path was the data.
Mm-hmm.
In other words, we wanted to, we followed the average data from these experiments. What is the average period? We want to get this period here.
Right. So I see you could shift the black curve downward and you would get all the other periods, but that extreme period at the right, you wouldn't get the very
That's
low.
correct.
Right. Okay, good. Yeah.
So that's what what drove us to do that. So it was the data. Obviously, you can choose other paths through this but we are trying to fit these data. And so again, you know, I've just simplified the pump equation so you can see the major players. I'm not giving you the components of the H current and leak, but just to remind you of the model. And now I'm showing you that same model as a single neuron. So this is what the single neuron does that came out of that half center. And again, it is rhythmically bursting, as you would expect in myomodulin especially. And you see the pump current and you see the internal concentration of sodium. And here the calibration is correct. The range of change of internal sodium we have is 1 millimolar. So we're not changing the internal concentration of sodium really a lot. It doesn't really do that. And this is a nice model because this model runs in real time on a signal processing board. So this is a real-time model, unlike the other one, which is a computer model. This is a real-time model, and it has this simplification that we're only looking at the major players in sodium. Okay, but it has all the other currents in it. It just doesn't calculate those. All right, so now what this inspired Ricardo to do is to try to restore robust bursting to isolated heart interneurons. So remember I told you at the beginning that if you take an interneuron from 7 in an isolated ganglion 7, that it no longer receives inhibition. And these neurons, even when recorded intracellularly, will burst weakly. But if we give them a little bit of P current and a little bit of artificial pump with a hybrid system, essentially dynamic clamp, then they undergo robust bursting. And this is shown here for two different levels of both the eye pump max and this persistent sodium current. So why did we did this because the currentscapes told us that the major contributor of sodium is the P current, is this persistent sodium current. So we wanted to look at how the pump and persistent sodium would interact with one another. And here you see the sort of the bursting that's restored with a GP of 4 nanosiemens and eye pump max of 0.2 nanoamps, and then you get much, much stronger bursting as you increase these the persistent sodium current. I don't know why I've got a reminder to come to this meeting, but I do. All right. All right, and then I wanted to look at this. So here, the robust bursting has been restored in an HN7, an isolated heart interneuron 7, and in the middle of this record, we see what happens is we are changing eye pump max. So we jump up in eye pump max, And when we do that, we see that the period of the system gets smaller, and the oscillations in sodium actually get smaller. So we're not here also working at saturation of the pump at all. In fact, the pump is working to keep sodium quite low when you increase I pump max. So that's just to show you that the pump can have dramatic effects on the bursting activity of these neurons. All right, now I wanted to go back and do some more historical stuff and talk about further evidence that the pump is actually important in mediating bursting activity in these neurons on a cycle-by-cycle basis. So this is work from Daniel Quay and Will Barnett and Genna and myself, and it's a bunch of different experiments here. Tell me if I'm going over time. So what was done here was to voltage clamp one of the cells here, and there's some escape spiking. We can't keep that from happening. We're voltage clamping at -45. Here's the voltage trace. And then we add the ionophore monensin. And the ionophore monensin increases the internal concentration of sodium. And you see that leads to a persistent outward current. So by increasing sodium concentration, we activate the pump. And then when we remove the monensin, then that turns off. Okay, and at the same time, that amount of monensin, if we didn't voltage clamp, would lead to a significant decrease in the period of the system. That, again, it's in a half center now, so you can see that. But if we block the H current, then you can see that the period change doesn't occur. But what happens is the burst durations decrease. And so the way we interpret that, and our model tends to agree with it, is that when you activate the pump with monensin, that helps to terminate bursts and also to provide hyperpolarization that activates the H current and allows the system to have a reduced period. But that when the H current isn't present, you just get one of those effects. You get the effect on burst duration. And then this is just to show that and you model the system by changing and putting monensin into the picture, then you get the changes in period that you would see under normal conditions. And the way monensin is entered into this equation here is as a term that just increases the baseline sodium concentration inside the cell. And the amount of monensin, and therefore the change in internal concentration caused by monensin, is expressed as a rate constant. And as you add more and more of monensin to the system, therefore more and more internal sodium then you get a decrease in period. And what was interesting here is that you see that the pump shifts, its current shifts upward as a result of changing the internal concentration of sodium, because the baseline sodium concentration moves upward. But if you were to just simply add this amount of current, this shift in current, then you don't get a change in period. In other words, the pump has to be dynamically active for this change in period to occur. So again, I think this is more indication that that the pump is very important in determining the burst characteristics of this system. And that's really all I had to say. And I'm open for questions, and I can go back to the presentation at any time for any slide that you would like to